and world-leading PSP and CBD researcher.
Good morning, everybody. Hello, it's very nice to join you all. I think being virtual on Zoom has advantages. We can have people all over the country, but it's not as nice as being in person, but we'll just have to make do. What I thought I would do in the first few minutes is to give an overview as to what I see as a way to see research into PSP and corticobasal degeneration as a pathway for beating these illnesses, really keeping that focus on the illness that people have. There's a role for mice and cells and all things in the laboratory, but the focus has to really be on this as a very, very human condition, and that brings some challenges and opportunities. So I'm going to bring up some slides on screen, which I hope everybody will then any moment now be able to see. Can you all see a big white slide saying beating PSP and CBD? Fantastic. First thing I'd like to say is that PSP and CBD, they're not really just one thing. There's a whole cascade of cause and effect. There's lots of things happening in the brain and in the body as these illnesses begin. We don't really know enough about what what I might call the root cause is, that ultimate cause. I put them in the blue box in the top left: genetics, environment, something else. We don't really know a lot about that, but we know much more about what's the next step in this cascade. That there's a buildup of this protein called tau. It's an abnormal protein. It's not the normal protein that we eat. It's something our body, our brain makes, and it starts to build up and get claggy and stuck together in cells. And with that comes a lot of inflammation in the brain. And we know quite a lot about this, and we'll hear more this morning about these two critical steps, because a consequence of that is that causes the brain cells to die back, to get shrinkage. They're spread through the brain. It starts in some very small parts of the brain and then over the years spreads out. And as it does so, it damages what are called synapses. These are the critical junctions, like all those wires and connections that plug our computer or phones together so things can talk. So those synapses go wrong. Even if the brain cells were alive, they can't work together. That's in that light green band of consequences. Beyond that, there's a particular problem I've highlighted in yellow called neurotransmitters. These are the chemicals that let the brain talk to itself, let the brain cells talk to each other. It's through a sort of language of chemicals in the brain called neurotransmitters. Many people may have heard of one called dopamine in Parkinson's disease. That's probably the best known of them, but there's many others. And many of these go wrong in PSP and CBD. And as a result of that, we lose what's called connectivity. The brain can't connect up to itself. It's like ripping out the wiring in the telephone exchange. Even if the wires are there, the messages can't get carried. And that leads to changes in movement, in cognition, sometimes behavior and personality, but very much more movement and balance at the outset. And all of these problems then ultimately lead to that, it's highlighting at the bottom right-hand side of the screen, that impact in terms of function. So QOL, that stands for quality of life. So in medical speak, I should have taken the acronym now, quality of life, and indeed survival. So as we come down this sort of rainbow cascade, there's lots and lots of causes and effects, lots of steps that lead up to the illness where we experience down the right-hand side there. And although it might sound complicated, one of the reasons I work from this framework is it gives many, many chances to stop or to slow down the illnesses. I think of each of these coloured boxes as like dominoes falling one after another. And for me, and I think for many of you online, what really matters isn't how much tau I've got in the brain. I don't know how much I've got. I don't care as long as my brain works. I don't even care how much neurotransmitters I've got as long as my brain works. So what really matters is not letting that last domino fall down on the bottom right-hand side, not having problems with movement, balance, thinking, not having an impact on our daily function or indeed life expectancy. So there's many, many ways to try and stop in this cascade, to stop those dominoes falling, to stop it reaching that last domino, the illness that's so troublesome. And I know, many of you online will experience at firsthand just what a difficult illness PSP and CBD can be. So this is all very much theory, but what's happening in practice? Well, this is actually, the way that the PSP and CBD world is moving. Now, I will say most of the trials are in PSP at the moment. I think that we'll start to see that changing over the next few years. I'll be happy to discuss in Q&A why CBD lies so far behind PSP. but most of the trials are in PSP. And I just thought I'd highlight a few of these to show how this cascade, this sort of rainbow cascade of cause and effect is helping our thinking. So at the bottom in yellow, there's the NORAPS trial. This is designed to replace a missing chemical in PSP called noradrenaline. It's the brain's natural adrenaline that's very important for all kinds of motivation, memory, thinking, and some aspects of movement. And it's very, very low in people with PSP on average. So we're using a drug to try and boost that noradrenaline level and see if that helps cognition, behavior, indeed possibly even in the long run, life expectancy. And we'll hear more about the NORAP study from my colleague Rob Durcan a little bit later on this morning. I think there's been some coverage of that in PSP Matters. What you might not have heard about in PSP Matters is those other two boxes in yellow on the right. A study from Alex Murley, funded by the Rose Tree Trust, looking to replace the missing serotonin that can be missing not in everybody with PSP, but in a high proportion. And when it's missing, it can cause problems with a change in personality, impulsiveness, irritability, and not so much movement, but many of the other more sort of deeply personal aspects of PSP. And that's been funded for a trial to start next year. I hope we'll be able to share more news of that once it goes live. And there's a study by Rebecca Williams and Laura Hughes and colleagues here in Cambridge, funded by Wellcome Trust, that's the big W, looking to replace GABA. GABA is another of these chemicals that's severely lost in many, but not all people with PSP. And we've been working on two ways to try and replace that GABA, but not replacing it if you don't need to replace it, trying to get the right drug to the right people, those who have missing GABA, which is by no means everybody. So a lot of work on neurotransmitters. You see in the top of the screen, Alex Murley, some of you may have met, but also Mara Malpetti that we'll be hearing from later this morning, who together with me and colleagues have two trials under the later stages of funding review. We're very optimistic about this now, of two trials to try and damp down inflammation, to slow the illness down. And the earlier we can treat that and slow it down, the better the prospects would be for people with either PSP or at risk of getting PSP later on. So those are all trials led by academics, by doctors and researchers in universities, but also our partners in companies are still active in the PSP space. I've highlighted on the left there a study called Prosper that's ongoing in the UK at the moment. There's one called Novartis, which is in setup at several UK sites and should get going with a vengeance next year. And of course, in the US, there's a big PSP trials platform that's trying multiple drugs simultaneously to try and halt PSP. So there's lots of activity, and I have to say this can't come soon enough to try and slow down or halt the illnesses. But all of this success in trials, getting treatments to bear, depends on lots and lots of work, much of which has been funded by PSPA in the last 10 years. I call it Know Thy Enemy, really getting to measure and understand what's happening in the body and in the brain with PSP, in human beings with the illness. We sometimes use the word biomarkers, which is anything that you can measure in a living person with an illness, what's happening on the inside. What you can measuring at each of these steps that you can see in sort of the rainbow cascade, but also investing in neuropathology. So understanding, being able to look for those who are willing to donate their brain to research, look in the brains of the real illness and PSP under the microscope to get to grips with the mechanisms and the causes. And we'll hear from a colleague, Annalise Quaggerbert, later on this morning, who's a real leader in the UK in neuropathology of PSP and how it can help leading towards better treatments. So the biomarkers, to measure, we can measure each of these processes in these rainbow boxes with different ways. PET scanning is particularly important for measuring sort of biological processes in the body and in the brain. We can look at fluids from the body, particularly spinal fluids, and we'll hear, I think, from David Rowe a little bit more about this. There's a whole world of blood tests, sometimes gets called proteomics, but basically this is just blood tests, measuring in the blood things that reflect what's happening in the brain. And this has leapt to huge leaps and strides in the last 5 years with ultra-sensitive assays, things that would have been unimaginably sensitive 5 or 10 years ago. A lot of work has used MRI scanning to measure the connections in the brain or to measure the chemicals that are missing in the brain and how the brain is sort of working and functioning as it's trying to think, read, move, and all those other things we like our brains to help us with. But also new ways to measure cognition and movement using what are called digital technologies. And I think PSP Matters, the magazine, has covered quite a few of these examples. Colleagues like Antonio Christolides in Oxford, for example, really developing new ways to quantify what's happening in PSP, because only if you can quantify it can you really help clinical trials prove that a drug is working. It's a sort of much under-recognized problem. How do you tell if a drug is working? One of the other things that's been very important the last few years is actually measuring the impact of the disease in ways that matter not just to what doctors see, but what patients and families experience. So there's a whole range of what we call rating scales that are really trying to focus on aspects of the illness that are important to people who have the illness or are supporting someone with the illness, rather than just what's been obvious to doctors over the years. So lots of ways to measure it. And with that measurement, I'm going to say know thine enemy and know thy researcher. So I'm just going to spend the last slide on highlighting who we're going to hear about this morning. I think we'll kick off with Rob Durcan. We'll talk about the NORAP study. He's been working in Cambridge for some years, recently moved back as a consultant in Ireland, but still very active with PSP and the research across the UK. Paul Baxter takes a slightly from Edinburgh. I've had a pleasure of working with in collaboration with Giles Hardingham and others. This has been a fantastic Cambridge to Edinburgh collaboration. I think the real heavy-hitting science led by the Edinburgh team, I have to admit, looking at how we can measure and protect synapses. I think that's what Paul will be talking about today. I actually haven't checked with him, but I hope so, because he's got some absolutely stunning work coming on how we can try and protect the brain from PSP and keep it working. Annelies Quaggeber is an academic pathologist working with the Brain Bank. And she's been very important in both in validating all these other tools and scans and blood tests. Does it really relate to the real thing, the real illness that's happening in the brain? But also in understanding mechanisms of the human disease. It's all very well looking in mice and stem cells, etc., but we really need to get to grips with the human illness. And the neuropathologist is key to that. David Vaughan, colleague at UCL, he could talk on many things. He's done a lot of work, important work around some genetic aspects of PSP, but one of the things I think he's made a biggest impact in the field of PSP is around new blood assays and tests, whether it's in blood, spinal fluid, or indeed in a skin biopsy, to try and pick up this mischievous tau protein. And it's a very special tau protein in PSP and CBD. It's different from other illnesses, and there's some new very sensitive tests that he's been developing and publishing with great impact. And finally, Mel Petty will end the morning. Again, she could also talk about many things, but what she's really, I think, becoming recognized, famous for, I might even say, but recognized for around the world, is in understanding inflammation and the role that plays in PSP and CBD, particularly because it's something that can be treated. So we'll see many different people working across the UK, in all parts of the UK, really trying to get to grips with many aspects of PSP, but all of them with that eye on better diagnosis and better treatments. And I'm looking forward to hearing what they have to say. Thank you, Nick. Back to you.
Thank you so much, James. A quick update before we kind of carry on with the session. Unfortunately, because Annelies is now on call today, she's going to speak at our next session, which will hopefully be held in February or March. So we're going to be joined by Professor Dan Smith from Durham University, who's going to talk about some Prism glasses work that he's doing. But I'll hand over to Rowena Ironside, who is our Chair of Trustees at PSPA, who's going to chair our first session. Oh, I just need you to unmute, Rowena.
Oh,
there we go. Thank you.
Morning, everyone. Thanks for joining us today. And my job really is just to introduce our speakers. So James has done a wonderful job of giving us an overview of what's going on and who we're going to be hearing from. And our first speaker is Dr. Rob Durcan. As James said, a neurologist. He's a PSP-funded research fellow who has been working in James's lab in Cambridge and has been key to the NORAP's clinical trials and its effect on symptoms of PSP, such as apathy and impulsivity. So is Rob with us, Megan?
Yeah, thank you, Rowena.
Yeah, he is.
Great. Over to you, Rob.
Megan, I've put a request through to share.
Perfect. You should be able to see that now.
Brilliant. Thank you very much. Good morning, everyone. I'm delighted to be here. It's a great honor to follow James always, talking about PSP, the ways we can treat it, and hopefully answers that could be available to you, your family, or anyone else who's affected by this dreadful illness. Thank you, Rowena. So as everyone has said, I am a clinical neurologist. I now work back home in Ireland, where I grew up and trained, but I spent 3 years working in James's group in Cambridge. And I delivered in a large part, my time there was in delivering this clinical trial using a noradrenaline boosting medicines. And noradrenaline, as James has alluded to, is one of these neurotransmitters or chemicals that are helpful helpful to us in daily life. So PSP is going to be the focus of my talk rather than cortical basal syndrome. So I'm sure many of you are well versed in this, but in essence, this falls under the umbrella of a type of brain illness which is called a Parkinson's plus syndrome. Okay, so it looks in some ways like Parkinson's, but it is actually very distinct and unique and has its own characteristics of symptoms, problems, features, and things that a doctor might find if they were to examine you. And it behaves a little bit differently to common Parkinson's disease. Often people have, through their journey of PSP, they have early on been told they have Parkinson's, but this is a very different illness, as I'm sure everyone here is well accustomed to. So common signs are falls, which are often very early in the illness. Your doctor might find difficulty with the way you move your eyes, slowness of the way your limbs move, or the way you walk, slowness of the way you speak or think.
Sorry, Rob, I don't want to interrupt, but we can't see your slides at the moment. We can just see your screen.
No problem.
Let's try again. There we go. That's better.
So changes in the way you think or in the way you behave. So the clinical trial NORAPS focused on really features of behavior, and so that's what I'm going to focus on. So the goals for everyone here who looks after or researches PSP is to find ways that we can find patients an earlier diagnosis and get them on a treatment strategy that's helpful. We have no evidence-based treatments that are proven to work. There's various drugs that we think do work, but really we need clinical trials to show that these work and to find better treatments. And ultimately, we want to improve the lives of patients and their families. So NORAPS is a clinical trial that boosts the noradrenaline in the brain for patients with progressive supranuclear palsy. And we're focusing on common symptoms which are seen in patients with progressive supranuclear palsy. So they're very common, but they're not necessarily universal. So we look at apathy. So apathy is the lack of motivation. And in essence, this is very common and it can be quite troublesome to live with. And it's losing your drive or your motivation to get up and do things. And often people who are who have a lot of apathy will be quite content to sit and do nothing for the day. And this is troublesome, as one can imagine. Impulsivity then is a change in the way you make decisions and weigh up the pros and cons when you're met with a fork in the road in daily life. And often people with PSP will not make very well-reasoned decisions, and this creates its own trouble, as again one could imagine. So apathy and impulsivity. So these are common illnesses in PSP, and they often coexist. And indeed, the more impulsive you are, the more likely you are to be very apathetic, and vice versa. So they seem to be strongly associated with each other. So as your illness moves on, often you are more impulsive and more apathetic. Okay. Why are these symptoms important? So patients who have more apathy, unfortunately, have a trickier disease course, and their illness appears to potentially move a little bit faster than people who are motivated. on a population level, they have earlier admission to care homes, or the loss of independence that comes with that. And there's a potential signal, but this isn't necessarily proof that they have a shorter time to survival with the illness. So if we can reverse apathy and give people motivation, there is every chance that we can improve their daily behavior, enable them to maintain their independence, enable them to maintain their position in their own home and a pipe dream. But it may well stand to reason that we could prolong someone's life by changing their behavior in life. Okay. Impulsivity then, so the kind of the thing that goes hand in hand with it of poor decision making, is very difficult for carers and families to live with. And some of you may appreciate this as quite a frustrating and difficult symptom to care for and to live with. And this creates a lot of burden from PSP, and this is a fairly major target that we'd like to improve. Okay, so the NORAPS clinical trial, when I looked at the various data points of people who were recruited into it, it's quite obvious that the more apathy and impulsivity that we see, the worse carer quality of life was. Okay, so this is really screaming at me that these are really worthwhile symptoms to aim to treat and to reduce. So if we can reduce apathy or impulsivity, there's every chance that it would downstream improve carer quality of life. And that's, like as I said at the start, a very clear target for us in the research community for this illness. So carer quality of life is a huge target that we want to improve. Okay, so we've established that these symptoms are worthwhile, and what is driving them. So apathy and impulsivity, both of these symptoms are associated with a lack of this chemical noradrenaline, the brain's adrenaline. And this is a chemical that we produce in a very tiny little part of our brain, in the bottom of our brain, just above your spinal cord. And this little factory of cells that makes noradrenaline, we all have one, but in people with PSP, this little factory of cells is devastated, and we see a lot of cell loss within that area, much more so than we see anywhere else in the brain, really. So if you compare them to people who of the same age who do not have PSP, this factory of cells can be reduced by up to 90%. Okay, so they're operating on potentially 10% of what a normal person who doesn't have PSP might be operating on with regards their brain's ability to produce noradrenaline. Okay, so they have not enough noradrenaline, and that may be strongly driving these symptoms that we have shown to be associated with not having enough adrenaline. And this is the tiny little structure at the bottom of the brain, that little blue spot. Okay, so that's what the area that makes the noradrenaline, and that's what we're interested on. That's the area that we're interested in. So this clinical trial really asks, can we improve these behaviors of apathy and impulsivity? And we used a medicine that's been around quite a long time, that's used for another illness called attention deficit hyperactivity disorder, and which is predominantly a childhood illness historically. But the way it works is really what matters. So it increases levels of noradrenaline in the brain. Okay, so it's an oral tablet, and it increases the brain's noradrenaline levels. It's a once a day tablet, and it's very well tolerated and safe in children. But we wanted to show if it is or is not effective in treating these symptoms, and then as a result, can we improve quality of life for the patient or their carers or family members. So the trial ran after a little bit of a false start in 2021. It ran from start to finish from 2023 to about a month ago. So it's now completed, and the last participant has completed the trial. And we're checking the various checks and balances and dotting the I's and crossing the T's on the data that we collected. And analysis is to commence very soon, with results anticipated in spring of 2026. And we look forward to that with a lot of eagerness and anticipation. So we'll see what the results are. So, and further streams of this research were focused on imaging features. So we encourage participants to have a special type of MRI scan that looked at that little blue dot that you could see on that brain scan. And this was encouraged, but not necessarily mandatory to participate in the clinical trial. And a number of patients, very thankfully, did undergo this MRI scan, and we're extremely grateful for them for doing so. So what this has shown is that there was an incredibly strong relationship between how small or damaged that little blue spot, the factory of cells where you produce noradrenaline, was, and how bad patient's PSP was, but also how bad their behavior was. Okay, so these are quite hard to really maybe wrap your head around, but these are graphs showing that the smaller your blue spot in the bottom of your brain was, the worse your PSP was likely to be, and in parallel, the worse your behavior of apathy and impulsivity was. So this is very reassuring and exciting to us as researchers, because this supports the argument for the trial that these patients who have more apathy and more impulsivity appear to have more illness in their noradrenaline factory, and therefore stand to benefit more from a noradrenaline boosting medicine. Okay, so this would support that NORAPs appears on the surface level to be an excellent idea. Another stream of this research is to look at blood samples which were taken periodically through the NORAPs trial. So at every visit, participants had blood draws. This would be 5 to 6 separate blood samples over 5 months, and this offers a very unique opportunity to look at biomarkers, these things that James mentioned earlier, which we can look at and see how did these behave over 5 months in patients with a clinical diagnosis of PSP. So that's an invaluable tool, and this is just getting underway now, and I think this is a very exciting avenue to look at if there's measures in blood which we can track over time which change, and potentially if the drug we're using in the clinical trial impacts any of these measures. So in blood we can measure hundreds of proteins, and these proteins give a measure of brain health, brain inflammation, or aging of brain cells. Okay, so a really exciting field, and I think the blood test is a very promising avenue of this research, and we're very grateful to PSPA for supporting it. What will the blood test potentially show? So if there are certain proteins which change in a particular pattern over time as your illness progresses, this might offer potential targets for future clinical trials, or it might offer potential biomarkers, so ways we can measure response to treatments in future clinical trials. So both are really worthwhile outcomes. In parallel, so I know I'm talking primarily about PSP, but Parkinson's disease, the cousin illness, which has some similarities, but importantly is a distinct and different illness. We're also scanning and taking bloods from a number of patients with clinically diagnosed Parkinson's disease to see, does this blue spot, noradrenaline factory, also undergo changes in patients with Parkinson's disease, and is there differences between how that finding expresses in Parkinson's disease versus PSP, and if there are differences in the blood measures that I've mentioned before. And this is useful to us as researchers to provide a comparator group for PSP. So if there's anyone on the call who participated in the clinical trial, I'd just like to express our eternal gratitude for the time you gave up, and I trust that it's been very much appreciated throughout everyone here. We're very grateful, and hopefully we'll have results that will change the way PSP is managed for yourselves or for the next generation of people who are diagnosed. PSP is a really difficult illness to live with, and importantly to treat, and we need to find better evidence-based treatments, and NORAPS is a stepping stone along the way, and hopefully it helps, and hopefully we've shown that. Early 2026 for the results. We look forward to them, and they'll be I anticipate made available at some point in 2026, and I would like to thank the PSPA for their very kind and helpful support in supporting me through my research time in Cambridge. I'm eternally grateful to have got the opportunity to do so. It was wonderful to meet so many fantastic people and to look after people through the clinical trial. But we're not finished yet. There's a bit more work to do. I'd be very keen to take any questions at all. If there was anything which was unclear, please do not hesitate to ask any questions. I'm very grateful for your time. Thank you.
Now, ask Paul Baxter to take the floor. Paul is a postdoctoral researcher at the UK Dementia Research Institute up in Edinburgh, part of the Geoffrey Young Research Programme, working on combining stem cell technology and high throughput screening methods to aid drug discovery. So welcome, Paul.
Thanks very much for having me. The classic Zoom question, can everybody see my slides?
We can, yeah.
Brilliant. There's a picture of me talking. Here's a video of me talking. Thanks very much for the introduction. Thank you again, Megan, for the constant invitation. I just thought I'd start by not actually introducing myself, but introducing the UK DRI, because I think it's very important. This is an MRC-funded, multi-university, multidisciplinary institute, so it's spread over 7 universities. I'm one of 1,000 researchers, and I think it's important because this has enabled us as researchers to really drive collaboration and cooperation rather than the competition that exists between us. And so what I'm going to talk about today has been a huge collaboration between the University of Edinburgh and the University of Cambridge, but there's more of us out there working on PSP towards this end. So about nearly 3 years ago now, we were introduced to the Geoffrey Young Research Programme, and this is named after an uncle, a father figure who died sadly of PSP. And they gave us some funding, and they identified a problem, which I'm sure we're all very acutely aware of, that there are no treatments that slow the progression of PSP. And they lay down the gauntlet, set us the challenge of how can we accelerate the development of disease-modifying medicines for PSP. And this is an incredibly difficult task, but I think with the power of the DRI working together with the clinical expertise and also patient experience and engagement, we can actually overcome this. And so this is very easy to say, difficult to do, but we do have a strategy. And this comes from the MND SMART clinical trials for MND. And this is the brainchild of my boss, Sanatan Chandran, and Professor Savanka Pal. And I could talk for at least 45 minutes on this at all, but I won't. What's really the two things I think that are incredible about this is that it's an innovation in how we do clinical trials. So you have, in a nutshell, you have multiple drugs. Rather than just testing drug X versus placebo, we have multiple drugs that we can take. And if a drug doesn't work, then we get rid of a drug, but we don't get rid of the patient. So this trial just continues and adapts. It feeds into itself, and it doesn't stop until we fix the disease. So I would love to see this happen for PSP, and that's really what I'm driving for. But I'm not a clinician. I'm a basic scientist. And one of the main tenets of why I think this is such an incredible way of going about clinical trials is the drug selection system. And it's this last bit, which I'll talk about today, which is using repurposed drugs in these high throughput screens of human models of the disease. Now, that's three incredibly large concepts, which I'm going to try and condense. So hopefully it doesn't get lost. And hopefully I don't say the word in a nutshell more than 8 times. So what I mean by drug repurposing, there are thousands of medicines approved in the UK and throughout the world. They'll be for cancer, for cardiac treatment. And essentially, instead of reinventing the wheel, we're going to take drugs that are already approved for use, that we know that they're safe for use, and see if they have any effect in PSP. And this significantly reduces the time from discovery of a drug to getting it into the clinic and treating people. And just, you know, from personal experience, the work that I was doing 3 years ago in collaboration with a big group into one drug, tacrolimus, that's now in a clinical trial with the MMD Smart. And as a basic scientist who started working with mice, that wasn't really something that I imagined would be possible for me in just such a short period of time. So how do we test these thousands of drugs, and how do we find out if they have any effect on PSP? The key part is you have to test thousands of compounds. So I brought myself some props, and it's moving from the standard in vitro way of using 24 well plates and moving to a 384 well plate, and really following an industry style pattern of working. And a lot of this has been really revolutionized for us, for collaborating with Neil Carragher, who's the professor of drug discovery at the University of Edinburgh, and he's formerly of AstraZeneca. And he's brought this, using this industry style methodology to working in cellular neuroscience. And, you know, with the way that we deliver, we store the compounds using these industry standards. And the caveat is that what we would do smaller scale, which would be very acute and very small tests, we need to do much more robust testing, much more robust cellular phenotypes. It's almost hitting it with a hammer as opposed to fine needlework. And that last part, using human stem cells, human models. This is a huge concept. It started off as a research from Ian Wilmot and Dolly Lashik in Edinburgh University, transformed by the Nobel Prize winner Shinya Tanaka. And essentially what we can do now is take stem cells from blood or hair cells, turn them into these pluripotent stem cells, and then by mimicking these signals that transform a stem cell into a brain cell in the embryo, so mimicking what happens in the embryo, we can actually create brain cells. And I can now grow neurons. These are human neurons with human astrocytes growing in a dish, and they will develop a network over time. I really encourage anyone who wants to know more about this to see Kat Bowles, who's a group leader at the UKDRI Edinburgh, who introduced this really wonderfully earlier on in the year in a PSPA session. So what are the cells affect? So we can create all of the cells of the brain now in a dish. What are the cells affected by PSP in the brain? The short answer is all of them. And just for time constraints, I can really only talk about the neurons. And I'm really fortunate being sandwiched between James and Maura at the end, because a lot of the core phenotype that we want to look at is really based on their work. And it's this finding that you have in PSP throughout the brain, you have a significant loss of synapses without, if you compare it to, say, Alzheimer's disease or FTD, Parkinson's disease, you don't have a huge number of dying cells throughout the wider cortex. And this is quite tightly associated with brain inflammation. So what are synapses? So the neurons are the rock stars of the brain. This is where all the information travels between cells and electrical communication happens. And at the synapse is where two neurons will meet and transmit chemical information across this bridge. And so you have a pre-synapse and a post-synapse, which are transferring information. And using my robot microscope, who I'll introduce again a little bit later, I can stain specifically the before side, the end side, and that's what these little dots are that are appearing on the screen. And so you can see that there are some yellow dots on the screen. That's where I have two neurons that are joined together, and they'll be transmitting information. And that's what's lost in PSP without, certainly within the cortex, without necessarily neuronal death. So how do we model PSP in vitro? This is very difficult. There are genetic changes that cause milder changes in developing neurons. Kat will talk wonderfully about this, about changes and mutations in tau. There's lots of work about GWAS that cause very subtle mutations. I think because these cause very slight changes, I think that's really why PSP doesn't manifest until much later in life. But they're certainly not the hammer blow, crayon-led, drastic changes that we need so we can readily adapt this to a high throughput strategy. So the way that we went about this, and this is really due to this collaboration with Cambridge, really led by David Kahneman here, is we took PSP patient brains that had been donated to the Edinburgh and Cambridge Brain and Tissue Bank. And David Kahneman takes these bits of brain, he extracts all the toxins that are in there. So it's not what's inside the brain cells, but the things that are being released that causes PSP to spread around the brain. We're removing these toxins, so tau and inflammatory cytokines. And then we can put this onto our neurons. And this gives us this unbiased way to see what the effect, the effect is. We're not just banking on cytokines to see what happens. And again, this is massively due to collaboration between all of the universities, Maria Spalentini, David Kahneman. Jane was instrumental in these cells and really directing the research. But above all, I'd like to thank the patients who donated their brains, because I think it speaks a lot to the bravery of these people who just refuse to admit defeat. When you put this isolate onto the human neurons, something truly incredible happens. And this was not something we expected to see. On the left, you can see yellow dots. These are healthy, happy neurons with lots of synapses. And on the right, after 24 hours of treatment, you can see this massive reduction in the number of synapses. It's like they're wiped out. And what is also incredible is you've got this loss of synapses. It's far more than if we were treating with tau. But you also have no neuronal death. And that's hauntingly familiar to what happens in cortex in PSP. So we decided that this was a phenotype that was strong enough for us to test at the high throughput level. And so we developed this assay where we could grow in these 384 well plates human neurons. We grow them for a month, and then add the compounds first. And we're adding compounds from these compound libraries, so they could have thousands or maybe just hundreds of drugs, but they're all either approved or depends on the biology that we want to, or chemistry that we want to chase. And then afterwards, adding this synaptotoxic PSP brain isolate. You can see that this is really sort of basic biology. It would be better if we added the PSP first, lost the synapses, recovered them, but we have to go with what's going to give us the most robust results. And in comparison to, say, other screens that you might do in cancer biology, we have to really upscale the number of repeats that we do in this assay, just purely to the heterogeneity of where you might see a synapse on a neuron. So this is an example. The last screen I did, 509 compounds, that ended up involving analyzing over 100,000 images. And that's only possible using robots. So I hope we can all see these videos playing. On the left, and this is this industry's level of doing research. On the left, we have a liquid handling robot, and that we actually inherited from the COVID testing center. And you can see that we can now use this to very strategically stimulate lots of plates with exactly the same amount of liquid, tiny amounts of liquid at the same time, keep it really robust. And then on the right-hand side is my robot microscope. And what this is able to do is take these thousands of images, and what's best is it has a robot arm, so that I can leave this over. Just due to that level of depth we need to achieve with the synapse screen, each one of these plates will take 18 hours to actually run. So we have to run this over the week, and that's why we need the robot arm to help us. And so this slide is 2 years' work put into one short slide. But I just wanted to include it to show you where we've come to. So over the last 2 years, I've screened over 2,000 compounds, and the last screen I'm particularly pleased with. It was a smaller, with a smaller screen designed to include drugs that were definitely CNS penetrant. One issue with screening lots and lots and lots of drugs that might be approved is that not all of them will go, when you take them, will actually go into your brain. Your brain protects itself from lots of the things that are going on in your body, and it has this membrane around it called the blood-brain barrier, which acts as a gatekeeper for toxins and so forth, but drugs in particular. So we use this specific chemical library that should pass into the central nervous system, and we start to see results. So if I go, here is 100%, the control number of synapses. You see this massive reduction of synapses here when we treat with PSP, and all of these hundreds of compounds, some of them are hitting above this line where we think, okay, this is interesting. The problem with moving into the high throughput screening is that you get a lot of false positives, particularly with quite a noisy assay such as this one. So you have to repeat it. You have to perform old school pharmacology where you do what's called a dose response. So increasing the concentration, the amount of drug that these cells see, and see if you get an effect that matches that increasing doses. And here I've simplified this by making green good, red bad. And you can see that not all of these are green, right? And they're not all green at the right-hand side. So we from about 30 hits, we've got 8 solid, following a dose response, positive hits that we believe are synaptoprotective. And it just comes down to having to be really robust with what we do. Now we've got these compounds, the next step is to then move it to a more complex model. So on the left here, we have an organoid. Right? Now these are like mini brains that we can grow in the lab. And what's brilliant about these is they're all human, again. We can introduce more of the cell types. So before I just had neurons and astrocytes in my plate. Here I've got blue neurons, green astrocytes, red microglia. We know these are important because of that inflammation in the brain. Microglia are like your white blood cells, your inflammation cells of the brain. And we can grow these for a long time, these 50 days, months, if we want to. And we can treat them for much longer. So we can, and then really ask a lot more complex questions with these than we could ask on the high throughput system. And this is very, very initial results. I probably shouldn't show it. But what we're finding is that using these 3D models, if I stimulate with the PSP brain isolate, we see the same loss of synapses. This was just a trial and error of going with one dose. We're starting to see some recovery with some of the drugs that we've identified. But more than just looking at what changes at the synapse level, we can start to do molecular biology. So looking at the RNA, this is an example of a qPCR. This is the signals from which parts of the DNA are going to be used at any one time. And by analyzing these, I can see what's happening to the neurons, but also what's happening to the astrocytes and to the microglia. And here it's just a heat map. Green is good, red is bad. And we can see that the PSP is largely causing this diseased state that's been recovered by some of the drugs, but not all to the same extent. So the next phase is we need to do more experiments. We need to test how effective these compounds are, and also I'm thinking back to Kat's talk again. Test how effective these are in the early phase genetic models, in the tau models. And there's a technique that we really want to do called single cell RNA sequencing. This tells us the molecular signature of all of these different brain cell types in response to the PSPI and in response to our drugs as well. And this isn't science for science's sake, because at the moment what we've got is a stimulus that we know is synaptotoxic, and we've got drugs that are synaptoprotective. But we really need to work on seeing if this effect is actually relevant to the PSP brain, to all of the different cell types there. Are there molecular signatures that we see in these 3D organoid models that we grow in the lab, and changes that we've observed in the PSP brain, post-mortem brains? And then what we can do, if we do see this overlap, we can then leverage this information to make more screens that can have an impact on the disease. And I think this is really important, because from what we've learned from the MND Smart is that you need to have multiple strategies hitting multiple cell types to really make impact on the disease, right? So we're not just going to have one wonder drug. We're gonna need multiple strategies and hit it with everything that we've got. So that's what we get from being able to get more screens. There's a huge list of people to thank. I'm just going to have to leave the list up, because this is really, it's been said many times, you need a village, but here we need a city of people to get this work done. But in particular, I'd like to thank the DEFRA Young Research Program for investing in us and driving us forward. And then a special thank you to all the PSP patients that I've met. I've been doing also a biomarker study, and I'd just like to thank you for inspiration and motivation for this work. That's me.
Thank you, Paul. Your excitement and enthusiasm for this approach really comes across, even if some of it is a bit a bit hard to follow for those of us lay people. It is your kind of excitement and enthusiasm that I think gives us an idea of the potential that you see in this approach to having multiple strategies and getting stuff done rapidly. So to our third speaker. So yes, I'd like to welcome Professor Dan Smith from Durham University. It's lovely to see we've got stuff going on all around the country now, which probably wasn't the case if we go back in time. So Dan's part of the Department of Psychology, and his research examines the interaction between the motor system, specifically the ocular motor system, you'll have to tell us what that is, and cognitive processes such as attention and working memory, with the goal of understanding how activity in the motor system helps resolve competition between different representations in the visual system. So over to you, Dan.
Okay, thank you very much, and thank you for inviting me to give this talk. Just to perhaps unpack the jargon that was there. Really, what I'm interested in is eye movements and how the systems that control eye movements interact with the systems that control thinking. And I'm particularly interested in attention and short-term memory. In this talk, I'm going to present. The last 3 years, I've been running a trial looking at attention and memory in PSP, funded by the Vivenza Foundation, what was the Dunhill Medical Trust. But I'm not going to describe, I talked a bit about that at the last one of these meetings I came to, so I'm not going to describe that research today. What I thought I'd do today is focus on a project I've been running actually with Megan, looking at more, I guess, therapeutic type approaches, looking at prism glasses. So I'm particularly, because that's of interest to me, as being interested in vision and the motor control and those systems. I'm particularly interested in to what extent do people with PSP actually find prism glasses useful. And I'm going to try and share my screen. I hope that will. So you can see my screen, I hope.
Yes, we can.
Okay, fantastic. So yes, I've sort of prefaced this a little bit. So this is work done over the last few years alongside our other work on that was funded by the Dunhill Medical Trust. I have some collaborators here in Durham, and of course, Megan has also helped a great deal on the PSP Association. Okay, so as we know, eye movement problems are very prevalent in progressive supranuclear palsy, and people with PSP often have problems making eye movements, and particularly in the downwards direction, and that's particularly true early in disease progression. So not all patients experience this, but many patients do. And if you have problems with looking down, that can cause a lot of other problems in just your everyday life. So it causes lots of problems with things like any task, really, where you have to be able to see that kind of workspace that's right in front of you. Sometimes this is called peripersonal space, but the space right in front of you where you would cut up food if you were eating, where you might hold a book if you were reading, or you might hold a mobile device if you were texting. So if you can't look down, it makes doing those tasks very problematic because you have to do things like hold a book right in front of you, right? Now, if you didn't have PSP, then you could easily compensate because you could just move your head down, right? You could actually shift your head, move gaze down, problem solved. But of course, people with PSP also have this additional problem where many people have rigidity in the back of the neck. And that makes it very difficult for them to compensate using head movements. So they're really kind of stuck looking forward. So because of this, one solution that's often been proposed is to use prism glasses. And I've got some illustrations of a type of prism glass called a recumbent prism glass. And I'll talk a bit more about it. But the idea is the light enters the prism glass here. I hope you can see my pointer. Bounces off a mirror that's placed on the top. And so it's reflected into the lower part of the eye. And so it shifts the image so that the area of interest now falls on the sensitive part of the eye so you can see what you're doing. And the idea is that this will be a very simple way to compensate for the loss of downwards gaze. I have my own set of prism glasses here. I don't know if you can see them with my blurring on. Put them on there. So now I can look straight ahead and I can see my hands. So I'll put them off. So these were actually developed in the 1930s as a visual aid for people with spinal injuries. So people with spinal injuries would be lying flat on their backs in their recumbent position, and Andrew McKee Reid developed these type of glasses to allow these people to be able to read more easily. As I said earlier, they're fitted over both eyes and they produce this global shift in image position. They're sometimes known as Hamblin prism glasses because the first manufacturers were Hamblin or Messrs. Hamblin and Company. If you look for them online, you'll often find them described as something like lazy readers or easy readers, and they're described as a useful device to allow you to watch television while lying in bed. So there are some good things about these type of prism glasses. So they have generally got a good optical quality. And what I mean by that is that you can see quite precisely what you're looking at. So the prisms don't distort the visual image too much. And they're relatively cheap. So you can get them for anything between 8 and 15 or 20 pounds. However, as we will see in a few slides' time, many people find them quite heavy, and they also obscure the straight ahead. So when you're wearing them, you can see what's in your immediate workspace, but you can't see straight ahead. Something else that Andrew McKee Reid originally envisaged them as being worn over existing spectacles. But many of the examples that you can buy online can't be worn over existing spectacles. They're just not designed to do that. And that might be a problem, particularly for older people and people who have problems that are just about how good their eyesight is if you need corrective lenses. So these recumbent prisms are the ones that are most frequently suggested for people with PSP. I should have a quick note to say there are other types of prism glass, so just to avoid any confusion. So what most people come across in terms of prism glasses are called wedge prisms. And these are glass or plastic lenses that are fitted into existing eyewear. And they produce very small changes in image position, and they are typically used to correct double vision. Right, if you've got a problem with double vision, you'll often be prescribed a peripheral wedge prism. There's also something called a Fresnel prism. So a Fresnel prism is a lightweight plastic version of the wedge prism. So what you have is, instead of having one very large prism, you have many small prisms in a row, and that can be fixed easily onto existing eyewear. So that's lightweight and it's cheap and can also produce a much bigger shift in image position than the wedge prism can. However, as you can see, actually just looking at these, you can see that there's quite a lot of optical distortion. So if you put this across your entire spectacle here, as in this case, that causes problems for things like reading because it makes the text somewhat indistinct. So the optical quality is not as good as the wedge prism, but they're much, much, much cheaper. Here's an example of a Fresnel prism fitted above and below on one side of a set of spectacles. So this type of prism is often used by stroke survivors who have had a visual problem. So many people with stroke lose the vision on one side of the world, and you can use these to expand the visual field. So it allows them to see things from their blind side. And in this case, they leave the center part of vision open to prevent double vision. Okay, so those are the sort of main uses of prisms. The question is then, are prism glasses useful for people with PSP? This is a key question, I think. And it turns out when we looked at the literature, there have only ever been 2 studies that have asked this question. The first one was in 1986 by Izzo and colleagues, who reported that prism users had positive outcomes. Okay, that is from the paper, but they didn't say on what tasks were measured. They didn't describe a protocol. It was rather unsatisfactory. The other study we found was by Sadowski and colleagues, or actually by Sadowski, I should say. And this was a single case study where they used a custom-built device. So this is prism glasses, and I don't know if you can see on the slide, they're mounted on a sort of arm here. And the idea is that the user can adjust the prism up and down for different types of tasks. And Sadowski reported that indeed this person with PSP was able to use the glasses to help recover some function for reading and find it helpful for eating as well. So that's it. There's 2 papers and in total, I think that's something like seven patients. So I think it's fair to say that very little is known about how many people with PSP use prism glasses. Do people with PSP use prism glasses? What kind of tasks they're used for? Whether they think they're useful, and what kind of challenges or hurdles people experience when using Prism glasses. And so that's what we set out to answer, is to start out with a very basic question, just get a sense of what's going on at the moment with Prism glasses in people with PSP. So to do this, we recruited 87 participants into a questionnaire study, and they were recruited in 3 phases. In phase 1, we recruited 55 participants, and they completed an online version of what we described as what we created, this Prism Use in PSP questionnaire. And we distributed this with the help of the PSP Association and also online and social media. In phase 2, we gave this questionnaire to 19 of the participants who took part in our Vivenza Foundation funded project. We had some more demographic details here. So the average symptom duration of these participants was 4 years, and on average they'd received their diagnosis 2.4 months, 2 years and 4 months, sorry, prior to participating. And in phase 3, which we completed in April, we had an additional 13 participants recruited directly via the PSPA, and they were sent either an online version or a paper and pencil questionnaire. And their average symptom duration was 5 years, and had received a diagnosis 2 years and 9 months prior to participating. Just one note: of our 87 people who completed the consent form and said they wanted to be part of the study, 12 of them completed fewer than 50% of the items, and so we excluded those people from our analysis. Okay, so what did we find? Well, the first question we asked was, well, have you even heard of Prism glasses? Well, I should say, so of our participants, 28 of them used recumbent prisms, and 10 were describing their experiences using peripheral prisms. So we asked specifically about recumbent prisms, but some people, it became clear in their answers that they were actually talking about peripheral prisms. So we split them into 2 groups. So of our sample, 71% had heard of Prism glasses. So the idea of Prism glasses seems quite well known. About half of the sample who had heard of Prism glasses had used them. Okay, so if you've heard of them, they're quite like to try them out. However, of the people who had used them, only 23% actually had kept on using them. So the majority of people who had tried using Prism glasses, and particularly the recumbent Prism glasses, had given up on them. So that's perhaps not terribly encouraging. The next thing we were interested in was whether or not essentially what the user's experience of using the Prism glasses was. So whether they found them useful, which is on the left-hand side, and whether they like them, which is on the right-hand side. So this figure just shows a cumulative percentage of all participants, and it says to what extent, how many of them agreed or disagreed with the statement, I found Prism glasses useful. And what we see here is that 15, so about, I think that works about 58% of the recumbent Prism users either disagreed or strongly disagreed that they found them useful. And in fact, only you can see 6 of the participants either agreed or strongly agreed. So again, there's not a lot of enthusiasm or not for using Prism glasses. The data for the peripheral prisms are here, but I'm not going to discuss them. It's not as interesting. I think we don't have too much time. So if we look at then the recumbent Prism glasses, do they like using Prism glasses? Again, a very large proportion either strongly disagree or disagree with that statement. So the take home here is that of our sample, only a small number of people find the Prism glasses useful, and an even smaller number actually liked using them. Okay, so we then asked them, what activities did the Prism glasses help with? So irrespective of whether you like them or not, did you find them helpful for anything? And what you can see here is just the percentage of the respondents that described these activities being helped by Prism glasses. And again, I've split this into a group of people who were reporting on recumbent prisms in this light grey color, and people who talk about peripheral prisms in dark grey. You can see it's quite striking that by far the most useful activity for recumbent prisms was eating. So very similar to what Sadowski showed in her study. Reading was also very frequently mentioned as activity that was helped with with recumbent prisms. And there's a handful of responses for other things like using tablets, computers, and things. For the peripheral prisms, again, they tended to be most useful for using tablets, using phones, watching TV, and reading. So people tend not to like using prisms. They don't find them useful. So we wanted to get a bit more detail about what was it they didn't like about using prisms, or what the problems that they experienced using prisms. So we had a number of different options that were described as that we allowed people to choose between. So things like heaviness, problems with glasses, self-consciousness, and these are all mentioned by quite a lot of participants. So the biggest problem with actually wearing with regular glasses, so the issue that I noted before at the start, and also weight. People also, prism glasses are not attractive things. They are very obvious you're wearing prism glasses. So many people talked about self-consciousness, and also issues around an unhelpful field of view. So something about the way that you were wearing the prism glasses, the way they were fitted, meant that they actually couldn't see the bits of the world they wanted to see. And so that's obviously a problem. Something else that might be an issue is side effects. So what kind of side effects do you get with using prism glasses? And what we see here is that disorientation was reported by a large proportion of the sample. So about a third of the people experienced disorientation. Other things included things like eye pain, double vision, and nausea. And you'll note here, interestingly, nausea was reported by a substantial minority of recumbent prism users, but none of the peripheral prism users. Okay, I feel like I've whipped through these slides. I've lost track of my timing slightly, but I'll just briefly summarize. Awareness of prism glasses as a potential visual aid for PSP was high. So around 72% of patients had heard of prism glasses. And we saw that they were most frequently used for, or perceived as most useful for, eating and for reading. However, somewhat disappointingly, only 48% of the respondents who had tried of the 48% of the respondents who had tried recumbent prisms, only 28% agreed or strongly agreed they were helpful, and only 23% continued to use them. So in terms of the kind of what you might call the clinical success, so by which I mean the measure of which whether people feel that this is a beneficial tool, prism glasses don't perform particularly well. And I said just 14% of recumbent prism glasses agreed they liked using them. 70% of users reported at least one concern with weight and problems wearing with existing spectacles, very common, and 40% of users reported at least one side effect, which disorientation was the most common. And indeed, 30% of users reported that prisms weren't helpful for anything at all. So how might we go about improving these outcomes? Well, one big issue is it takes practice to get used to wearing prism glasses. So when we use prism glasses in the lab, what you need to do is give your participants normally 5 or 6 minutes worth of practice to get used to the fact that the visual image has changed, and now when you reach to something, it's not where it used to be. So you have to effectively basically recalibrate your visual motor system. So you have to learn how to use the new visual input. But none of this kind of information is typically provided to patients, particularly those that get it via somewhere like the NHS or read about it on a website. They just say, oh, try prism glasses, but with nothing else. So one very low-hanging fruit is potentially just to provide a bit more information about what you need to do once you're wearing the prism glasses to get used to wearing them, and a bit more information about what kinds of things might benefit from prism glasses. So we saw that things like eating and reading, yeah, they seem quite successful. Watching television, not so much. So, maybe a bit more what you might think of expectation management about how you might use these glasses. A caveat to that, of course, is that PSP is a movement disorder, and in order to practice wearing the spectacles, you have to reach to grab things. And it might be the case that the very fact of PSP being a movement disorder makes it harder for them to adapt to wearing the spectacles, right? Because the motor and cognitive systems that you use to adapt to wearing spectacles are the systems that are affected by PSP. And there is some evidence, in fact, that people with Parkinson's take longer to wear in prisms than older people without Parkinson's disease. So it might be that we do need to do some sort of more specific training. Another caveat is that our participants were relatively late in disease progression. And so many of them may have had oculomotor problems or problems with their eye movements in many directions, as well as up and down. And that itself might make a set of glasses that you can only change vision for downwards gaze less useful. So perhaps Prism glasses might be more useful earlier in disease progression, or specifically useful for people only with downwards gaze problems. And so we might also think that customizing Prism glasses might be necessary for more optimal outcomes. So using lighter weight materials, finding better ways to fit them, or even having high levels of customization where you can move the prisms around a bit like in Sadowski's paper. So that's it. Those are the kind of message I wanted to get across, the results I wanted to share with you. So I just want to say a very big thank you to the PSP Association for supporting this research, and of course to all the participants who took time to fill in the questionnaire and send them back to us.
Thank you, Dan. Very, very interesting. And I think identifying that one of the things that might make these devices more useful to people is just providing a bit more information is something, you know, that we can really at the PSPA do something about. So yes, that's an actionable item, which isn't always the case in the outcome of the research.